Bienvenue à bord. Welcome to the fast curiosity of MechaCuisine. Today we're going to venture into the question, why salt makes things sweet? This is potentially an irony, because normally the question would be why salt is bitter. But there's a twist here. In the comfort of your kitchen making some cookies, you notice that salt is one of the ingredients. So is it a bittersweet oxymoron? But here's a secret that kitchen engineers or rather food crafters don't tell you. Brain trick. They actually make you think your food is sweeter by adding a tiny little pinch of salt. So naturally in our tongue taste buds, we have a cell called a T1R cell, which basically perceives both natural and artificial sweetness present in the mouth, sends signals to the brain to imply a feeling of pleasure. T1R cells are classical receptors. With a glucose touch, they trigger a chemical signaling cascade, telling your brain this is sweet. However, there's another protein called a sodium glucose cotransporter, an SGLT. It doesn't just pass glucose to the T1R receptors, it provides an alternative pathway for sensing. An SGLT pulls glucose and sodium into the cell, changing the electrical charge, depolarization. This electrical shift triggers the cell to fire a signal to the brain, even if the T1R receptors are blocked or saturated. Then we'd ask ourselves, isn't it more of the velocity of perception? No. Apparently not. SGLT does increase sweetness. When sodium is present, it causes the SGLT to provide an additional signal to the brain that adds to the signal from the standard T1R sweet receptors. This makes the same amount of sugar feel more intense, which is why a small amount of salt can make sweet taste significantly sweeter, like 2 peas in a pot. And SGLT1 is primarily a benefit because it's a high affinity sensor, meaning it can detect glucose at much lower levels than the standard T1R receptors can. This makes your perception more effective because you can taste or sense sugar even when it is too diluted for your main sweet receptors to pick up. Speed versus intensity. While SGLT does not necessarily make the conscious thought of this is sweet happen significantly faster in your mind, it does trigger a rapid physiological reflex. For example, an SGLT on the top helps trigger cephalic phase insulin release, a preemptive rise in insulin that occurs within minutes, long before the sugar is actually absorbed into the bloodstream. So it's a trisaccharide protein, which does not only increase intensity, it also has high sensitivity and metabolic efficiency. Let's do an electrophysiology summary. So let's take the sweet taste cells as the battery, and this battery requires an electrical charge to send a sweet signal to the brain, like flipping a switch, depolarization. The SGLT is a sweetened duck the moment sodium isn't present, but the moment sodium comes in the picture, the SGLT pulls the sodium together with the sugar molecules to the test cells, and this cell becomes depolarized much faster or more intensely due to the high concentration of sodium ions, and because of the test cell firing more aggressively due to the electrical kick, your brain interprets the signal as a stronger sweet sensation. So this whole process takes place much more faster and more intensely than sugar can do on its own with the T1R receptors only. The chloride ions do not necessarily disappear in this picture. The chloride is rather not transported across the membrane by this protein. Instead, it binds to the outside of the protein. It changes its shape to make it easier for external sodium to bind. Chloride is a helper. Stays on the surface to keep door open for sodium ions and sugar. Today we have deconstructed a biological glitch. In engineering, we would term the SGLT as sensors. They're picky eaters, only moving glucose into your cells if sodium is there to open the door. Think of it as a mechatronic system. The salt is a system override key. When a tiny bit of salt hits your tongue, it triggers the sensors to work overtime, sending a super sweet signal straight to your brain's processing unit. Note, too much salt causes system failure. It tastes salty, but just enough acts as a signal booster for sugar. That's molecular gastronomy, where chemistry meets the kitchen. Explorons la science as sample. From circuits to cupcakes, the logic is the same. I'm engineering the kitchen of the future, one molecule at a time. Stay curious and keep hacking the recipe.